induction of mitophagy in the HEI-OC1 auditory cell line and activation of the Atg12-LC3 pathway in the organ of Corti. Source: Hearing Research. Authors: Cristian, Setz, Anne-Sophie, Benischke, Katharina, Pinho, Ferreira, Bento, Yves, Brand, Soledad, Levano, Franziska, Paech, Katharina, Leitmeyer, Daniel Bodmer. Autophagy is a highly evolutionary conserved quality control defense mechanism within cells, which has also been implicated in cell death processes. In the mammalian inner ear, autophagy has been shown to play a role during early morphogenesis as well as in adult cochlear hair cells exposed to ototoxic insults. Mitophagy, a selective autophagic cell process targeting mitochondria, hasn't been studied in the inner ear so far. On this work, we searched for molecular indicators of mitophagy within Helsier Institute organ of Corti 1, HEI-OC1, cells as well as in the organ of Corti, OC. We first tested for the expression of Pinch1/Part 2 mRNA in 5-day-old C57BL/6 mice's cochlea using RT-PCR. We focused on the induction of mitophagy in HEI-OC1 cells as well as in the OC and investigated a possible mitophagic potential of the aminoglycoside agent gentamicin. The induction of mitophagy in HEI-OC1 cells was detected by objectivizing the translocation of fluorescence-tagged LC3 to mitochondria using confocal microscopy after a 6-hour incubation with a well-described mitochondrial uncoupler and mitophagy-inducing agent, carbonyl cyanide m-chlorophenyl hydrazine, CCCP. Incubation with gentamicin generated no mitochondrial translocation of LC3. Protein levels of COX-IV, Atg12 and LC3 were evaluated by an immunoblot analysis after a 24-hour CCCP treatment as well as gentamicin. We demonstrated mitophagy after CCCP exposure in HEI-OC1 cells by showing a downregulation of COX-IV. A downregulation of COX-IV could also be visualized in the OC after CCCP. A significant oxygen consumption rate, OCR, changed in cells treated with CCCP as well as significant morphological changes of mitochondria by electron microscopy, strengthen this assumption. Gentamicin exposure generated no impact on OCR or mitochondrial morphological changes by M. Finally, we demonstrated changes in the expression of Atg12 and LC3 proteins in both the OC and HEI-OC1 cells after CCCP exposure but not after gentamicin. Our data indicate that gentamicin had no impact in the activation of mitophagy, neither in the HEI-OC1 cell line nor in the OC. Therefore, we speculate that mitophagic-independent mechanisms may underlie aminoglycoside ototoxicity.